Okay. Hi guys, I'm Tuba. I'm Yara.
I'm Aaron.
I'm Mihir.
I'm Jose.
And we are BioMe, and we're very excited to present to you our novel gut on a chip system. Let's go over some key concepts about what gut on a chip system is. As you guys know, it's a micro-engineered system that basically aims to recapitulate the gut, basically on a chip. It consists of transparent 3D polymeric material, usually, that is lined by human cells. And then most, based on past work, we know that most gut on a chip systems try to replicate three important aspects of a gut. First is the 3D microarchitecture, which usually includes including the microvilli, as one of the teams here has done. That's usually done by either creating the actual shape of the microvilli or either inducing peristalsis by vacuum chambers. Another aspect is functional tissue-to-tissue interface, which involves changing, for example, the permeability between the blood vessels and the gut tissue. And you can do that by manipulating, for example, the materials that you use. The third aspect is changing the gut-specific biochemical microenvironment, which includes exposing the gut cells to specific metabolites or nutrients that are usually present in the gut. So why do we use gut on a chip systems? We know that we can use it as a diagnostic tool to better understand, for example, how the human microbiome is linked to specific diseases. We can carry out controlled, repeatable experiments, just because how easy or quick it is to make these devices, and essentially it reduces the need to have animal testing or human testing, just because having the actual cells of, for example, a patient within a gut on a chip system can allow for more accurate testing, and it can be more patient-centered. And it also is a platform for drug screening and testing, which is currently being developed by a lot of labs these days. And despite all the progress that's been made in this field in the past few years, there are some unmet needs within this field. And so our team was very focused on kind of looking and honing into these unmet needs. And instead of reinventing the wheel, we really wanted to kind of pick a specific small problem and try to fix that. So the first one that we obviously noticed, similar to other teams, was that a lot of the devices that were being made in this field were made out of PDMS, which, as we understand, is permeable and is not the most accurate way to test drugs. So our original idea was to use a resin called Prothresia, which is basically very similar to PDMS, but is 3D printable and is non-permeable, which we thought would be a great platform to make a gut on a system that could be efficient for drug testing. However, we ran into some issues that Jose will go into later on, which is why we decided to, at some point, change directions, and we went from the second aspect to the third aspect of recapitulating a gut system, which is to change the microenvironment and try to mimic the microenvironment of the gut, basically. And essentially, a major thing that, as we understand, is that the microbiome of the gut is an important and very essential and integral part of the gut that affects a lot of things. However, in a lot of new devices that are coming up, it's been hard to co-culture anaerobic bacteria with the CACO2 cells. So we wanted to create a system where we basically changed media and allowed our CACO2 cells to grow without glucose. One of my team members, Mihir, will go further into how we changed directions and how we've been carrying out experiments to further look into this new aspect of recapitulating basically the gut. Now we'll go into more about the design choices that we made. Aaron will now present all the design choices that we made for our system.
Okay, so our model for inspiration is the Kim H et al. Model. So this is basically like a strict outline of what we have, and then so this right here is our initial CAD for our initial design, and so it basically is based off of the guidelines outlined in the Kim model. We have a main chamber that is right there, right in the middle. We have inlets and outlets outlined by the circles there. And then we also have vacuum chambers that surrounded the main chamber on both sides. So talking a little bit about functionality and what exactly we wanted to get out of our device. So this image here is like a cross-sectional area of the device looking into the channels. So basically within the main chamber, we would have, this would contain media and CACO2 cells. And so this is where we would establish the epithelial barrier. the two chambers, so the top and the bottom one, are separated by a PEEP membrane to mimic the physiological conditions of the gut. And then on the sides, we have vacuum chambers where we could basically move air through them to establish the peristaltic motion of the gut. And so to talk a little bit about our fabrication process and how we basically started assembling these devices. So we started off with that initial CAD, which over time we refined and made modifications. But basically we started off with the CAD, we would print a mold, and then using that cast, we would put PMS in there, cure it, and then take out the molds. So as I mentioned before, we used a PEEP membrane to separate the two channels. And so at first we realized that they wouldn't really bond to the PMS, so we had to use APTs, which would help make them bond a little bit better. We used PDMS casting, and then to basically build the device, we would plasma treat one of the layers onto the glass slide, and then plasma treat the other complementary layer onto the other PDMS. So along the lines, we encountered many fabrication and assembly issues that we had to fix basically through trial and error. So one of them is that at first the 3D printer wasn't producing the cast that we needed, so that kind of set us back with starting off with our initial devices. Within plasma bonding, there were a lot of issues there. So one of the things that we noticed is that there were some hazy areas that we noticed in some of the devices, and so this is where the PDMS did not properly bond. And so when we would do like flow testing, we would notice that there would be like seepage around the channels and stuff, and so obviously that would cause problems there. One of the problems that we also had was trying to like align the layers correctly. And so obviously this would also cause like flow testing issues as well. And then just throughout the process in general, there would also be like dust and particles that would accumulate on the PDMS. So sometimes that would cause clarity issues. And then specifically with the APTs membranes, sometimes what would happen is that they would bond to each other, and so that made it difficult to like cut them out and like produce them.
So yeah, after a couple of iterations with our first designs and testing with food dye, we decided that we needed to change a couple of our design of our cast. So it was advised to us that we should remove the vacuum chambers because some of the media would flow into that because it wasn't properly bonded, the two PDMS. To improve the alignment of the two channels, we included alignment markers at the top, which is similar to other teams, which are just crosses. And we also just increased the height of our channels because our initial design would only hold about 1.5 microliters, which would be very unfeasible to like have a high concentration of seeds. So we decided to increase the height to make it easier on us to meet the cell density that we wanted to have. Because we had to remove the vacuum chambers on our devices, that sort of we lost the ability to sort of mimic peristaltic motion, but we do have the ability to add flow to our devices to sort of still mimic that dynamic environment, because that's sort of an essential thing when wanting to model the small intestine. So some things we did to in our initial steps to test how well our devices are. First, as everyone has done thus far, dye testing, get some colored water and just see how well the devices bonded to each other. As Aaron stated before, when we had the PEEP membrane and the two PMS bonded castings, we had a lot of leakage, and that's because the PEEP membrane by itself would not bond to the PDMS, so there was like a lot of gaps there, so that's why we had to use the APTs treatment. As well as we wanted to just see whether or not there were some cells in our devices, just using, as everyone, the confocal microscope. And some other tests that we want to see is just after a couple of days, is our device good for cell viability? So some problems encountered as stated before. So prothresia seemed to be a very difficult material to work with. It was difficult to obtain, and when we were talking to a collaborator that wrote a paper on prothresia, the device that we sent her was too big for her manufacturing process. We had a lot of trouble maintaining our cells. There was a lot of confluency at some point. There were some cases where our cells became contaminated. There were other cases where our cells just weren't there. When we were trying to view our devices on the confocal microscope, it was made apparent that our devices were too thick in order to see into the devices. So in future iterations, we need to either decrease the thickness of our PDMS or find a different method of viewing these cells. And as stated before, our channels are still relatively small. It went from holding 0.5 microliters to holding about 2.5 microliters in each channel. It's still very difficult to get a very high cell density to get into our devices.
So finally, after we got our final design to work, we did the dye testing, and then we wanted to start seeding our cells. So earlier this week, we started adding the cells to our actual device. So far, they've been seeded in 3 devices for 2 days, and the media has been changed. We seeded 15,000 cells, which we've been told might actually be too large of a number, so we're kind of going to be playing around with that. We got that number from the Kim paper that we are modeling our device off of. So we're going to hopefully see whether or not we should decrease the number since they grow so rapidly. Yeah, and then so earlier we also tried to stain the cells. However, we had the problem with the imaging since the device was so thick. It was so hard to see like whether the cells were there or not.
All right. So another set of experiments that we've been doing simultaneously is using transwells. So these are just small little devices that you can put inside a 24 or 12 well plate. And what we are trying to see is the effect of how the media affects whether we can grow cockatoos and whether we can use that for a future application in microfluidic devices. And specifically, the idea that we were going off here is that the media is going to affect greatly the metabolism of the cockatoo cells. In particular, we saw this paper that I've attached below. It's by Jansen, Houston et al., and that showed that that was actually testing a different question. That was seeing how if you deplete ATP in the cells, the cockatoo monolayer breaks down. However, what we got from this paper also was that if the cockatoos are cultured in a galactose-containing medium instead of a glucose-containing medium, the cells tend to rely more on mitochondrial ATP production rather than just glycolysis, which is a feature of most cancer cell lines. And so the idea is that we culture these cockatoos in different combinations of media. So at the top we have 100% BioMe glucose, the standard media that we've all been using. And at the bottom, as we go down the rows, we can increase the percentage of galactose. So here we have 50% galactose by volume, 50%, and then 75%, and then 100%. And we seeded these cells about 6 days ago, and they're approaching confluence now. And over the next week or so, we plan to test for metabolism and viability using Presto Blue and possibly TIR. And here's just some pictures that we captured from the scope on our transwells. We can see that the cells have been growing, and even without glucose, the cells have been doing fine. And so we think that is promising for future device. All right, and then so our novelty comes with the transwells and the idea of using a different media than just standard BioMe glucose. And so that will take a few more steps to implement.
Yeah, we also have novelty in terms of the device design. So as mentioned earlier, we are using actives to treat our membranes in order to create better bonding between the PDMS and the membrane. And then our membrane choice, we have polystyrene, courtesy of BioJax. Thank you guys. So that just makes it significantly easier for us to image into our devices. Also regarding alignment, so we mentioned we have the alignment markers. So at the moment, even with those, there's so much room for human error in terms of the placement of our devices. So we're currently coming up with like a mechanism in order to better like place them or place the 2 class or PMS layers on top of each other. So that should be a spot for now.
All right, and we engaged pretty rapidly with the patent process. At the beginning of the semester, we got the first 2 LM patents in. We sort of built off the second one in particular with our transwell experiment. We are trying to use galactose instead.
So conclusions in what's currently happening in the field. So there's a lot of engineering challenges. Something that a lot of teams were trying to tackle, and we also tried to start with, was really like material selection and trying to choose the best materials. So for many reasons, PDMS obviously is not the most ideal, but we did stick to it. So that's a challenge for the future. Reusability. So we realized that every time we created a new device, if we like put dye in it to test it, suddenly we couldn't use that device. So you're just kind of like burning through things very quickly. So coming up with a method for us to be able to reuse that same system. And then guaranteeing device integrity. So like when I test a device and see that the food dye works or like doesn't seep through on this one, does that necessarily mean that my other device also will be fine and I can also add media to that? Especially with our issues with alignment. So one device might be aligned more correctly than the next. And then we just need to come up with a better way to make our devices more reliable for widespread use in the future.
All right, so if we were to have a few more months to work on this, here's how we might go forward with this. So with materials, we agree with the other teams that PDMS is not the optimal choice going forward with microfluidics. And so we'd like to go back into looking at compatible materials and figuring out how those can be used in our devices. And as Yara mentioned, we need to be able to have a dependable device that we can be pretty certain of that will work. And we shouldn't have that device-to-device sort of variability. And we would continue with the transwell experiments. We try to track the cells over a longer period of time as they become more adapted to their media. And hopefully this will be an iteration towards co-culturing anaerobic bacteria with pathogens.
So advice to future researchers, it's really important to focus on one major problem. I mean, there's so many things that you can tackle currently in the field, but once you're trying to tackle so many things, you're creating too many variables that are interacting with each other. So it's just easier to focus on one main thing, and that's kind of like the methodology we went with. We started with the material, and that didn't work out, so we found a different route. There's still lots to learn about in the field, and then we really tried to use the mentality of like failing fast and iterating. So as much as we wanted to stick to the material, we realized we only had a semester, and we wanted to have some type of data by the end of it. So we very quickly were able to switch our project and try something new.
At the same time, yeah, we definitely said go into the lab. This is our space, so let's make the most of it. Let's get all the skills that we can get, and also let's collaborate with the other teams. We're competing, but at the same time we're collaborating together, and let's build off each other and go off over. All right, and so, yeah. Right.
Bye.
BioMe. All right.